Hello, my dear students. Today we will talk about another different processes that take place within the cell membrane. These are active transport, endocytosis, and exocytosis. Our key concept for today: cells use energy to transport materials that cannot diffuse across the membrane. We already know what is diffusion, and it does not need energy to pass molecules in and out of the cell. So today we will talk about the molecules that need energy from the cell to transport these materials in and out. Our main ideas: number one, proteins can transport materials against a concentration gradient. We already know the meaning of the concentration gradient. Number two, endocytosis and exocytosis transport materials across the membrane in vesicles. So let's start. The first one, proteins can transport materials against a concentration gradient. You just learned that some transport proteins let materials diffuse in and out of the cell down a concentration gradient. Many other transport proteins, often called pumps, move materials against a concentration gradient, and this is obvious why they are called pumps, because they are moving the materials against the concentration gradient. So what is active transport? Active transport drives molecules across a membrane from a region of lower concentration to a region of higher concentration. So this is the opposite of the passive transport. Okay. Active transport uses transport proteins powered by chemical energy because it is pushing the molecules. As you can see in this graph, the molecules concentration outside is lower than inside. And although this concentration, the molecules are moving from the area of lower concentration outside the cell to the area of higher concentration inside the cell using energy. Cells use active transport to get needed molecules, regardless the concentration of gradients, and to maintain homeostasis. And we already know that homeostasis is the stable internal environment of the cell. All transport proteins span the membrane, and most change shape when they bind to a target molecule or molecules. Some transport proteins bind to only one type of a molecule. This is like the enzymes, while others bind to two different types. Some proteins that bind to two types of molecules move both the molecules in the same direction. Others move one molecule in one direction and move the other molecule in the opposite direction. The key feature of active transport proteins is that they can use the chemical energy to move a substance against a concentration gradient from lower concentration to higher concentration. Most use energy from a molecule called ATP, and we will study the ATP later, adenosine triphosphate, either directly or indirectly. For example, your nerve cells or neurons need to have a higher concentration of potassium ions and a lower concentration of sodium ions than the fluid outside the cell. So in this case, there is a sodium-potassium pump. The sodium-potassium pump uses energy directly from the breakdown of ATP. It pumps 3 sodium ions outside the cell for every 2 potassium ions it pumps in. Another thing is the proton pump, is another transport protein that uses energy from the breakdown of ATP to move hydrogen ions or the protons out of the cell. And here is the sodium potassium pump. As you can see, this is within the cell membrane. For every 3 sodium ions outside the cell, it gets 2 potassium ions inside the cell using the energy of ATP directly. The second one, as we said, another example is the proton pump. The transport proteins, another transport protein that uses energy from the breakdown of ATP molecule to move the hydrogen ions, which are named the protons, out of the cell. Okay, this action forms a concentration gradient of hydrogen ions, which makes the fluid outside the cell more positively charged with these ions than the fluid inside. In fact, this gradient is a form of stored energy that is used to power other active transport proteins. In plant cells, this gradient causes another protein to transport sucrose into the cell, an example of indirect active transport. So this gradient in plant cells causes another protein to transport sucrose. This is a sugar into the cell. So this is an example of indirect active transport because it will get the ATP molecules but in an indirect way. A cell may also use energy to move a large substance or a large amount of a substance in vesicles. Now we will be talking about endocytosis and exocytosis. Transport in vesicles lets substances enter and exit the cell without crossing through the membrane. Let's see. Let's talk first about endocytosis. Endocytosis is the process of taking liquids or fairly large molecules into a cell by engulfing them. Engulfing means swallowing in a membrane. In endocytosis, the cell membrane makes a pocket around the substance. Then the pocket breaks off inside the cell and forms a vesicle which then fuses with a lysosome or a similar type of a vesicle. Why a lysosome? In order to break down this pocket or this vesicle and also break down the materials inside if the cell needs to. Lysosomal enzymes inside the lysosome break down the vesicle membrane and its contents if necessary, which then are released into the cell. As you can see in this GIF, these are the materials that the cells need to take inside the cell. So the cell membrane form a pocket around these materials and they get into the cell to stick to a lysosome and the process begins. This is an example or this is an illustration of endocytosis, taking some materials inside the cell. Okay, what about phagocytosis? We have phagocytosis. If you can see this red particle on the picture to your left, so this is like a pathogen, and the green one like the white blood cells. So it engulf or phagocytosis means the cell eating. It eats the whole part in order to damage it. Can you guess how the white blood cells damage these parts? Yes, for sure, by the enzymes inside the white blood cells. Also, can you see this black particle? This is under an electron microscope, the one to your right. This is one of our defensive cells in our white blood cells. This is a cell trying to phagocytosis or engulf a foreign particle to get rid of it. So phagocytosis is a type of endocytosis in which the cell membrane engulfs large particles. Phagocytosis means the cell eating. Phagocytosis plays a key role in your immune system. Some white blood cells are called macrophages. Macrophages help your body fight infection. It finds the foreign materials, such as bacteria, engulf them, then destroy them using the enzymes. So this is endocytosis. Let's talk about exocytosis. Exocytosis, the opposite of the endocytosis, is the release of substances out of the cell by the fusion of a vesicle within the cell membrane. During exocytosis, a vesicle forms around the materials to be sent out of the cell. Then the vesicle moves toward the cell's surface, which is, yes, for sure, the cell membrane, where it fuses with the cell membrane and lets go of its contents. Look at this one to your right, the materials. There is a vesicle around the materials to be sent out of the cell. Then the vesicle moves to the cell membrane, fuse with the cell membrane, then open to the outside, releasing the materials to the outside. This is the exocytosis. Number one, the vesicle inside the cell, it moves to the cell surface, which is the cell membrane, fuse with the cell membrane, then open to the outside. Exocytosis happens all the time in your body. In fact, you couldn't think or move a muscle without exocytosis. Let's see an example. When you want to move your big toe, for example, your brain sends a message that travels through a series of nerve cells to reach your toe. Okay, this message or nerve impulse travels along each nerve cell as an electrical signal, but it must be converted to a chemical signal to cross the tiny gap that separates one nerve cell from the next. Yes, we already know that. Our nerve cells are not connected in one line. No, there are very small, tiny gaps in between each cell and the next one. So the electrical signal, it will travel through the whole cell. Then when it reaches this gap, it needs to be transferred to a chemical signal to cross the gap, then be converted in the next nerve cell into electrical signal again. Let's see. Then these chemicals are sorted in vesicles within the nerve cell. So this is the nerve cell to the left. It have the electrical impulse, and it says, or it orders the vesicles to get out of the cell. This is exocytosis. When a nerve impulse reaches the end of a cell, it causes the vesicle to fuse with the cell membrane and release the chemicals outside the cell. Then these chemicals reach to the other nerve cell. Then they attach to the next nerve cell, which triggers a new electrical impulse that travels through the next cell. I hope you understood this part, exocytosis and endocytosis. See you in the next episode. Have a nice time and thank you.